Start with Chapter 7, reflex arcs and the use of the computer to demonstrate neuron function. Going back for a little bit of review, the electrical activity in the neurons. Let me see if I can get this slideshow to come up a little bit better here. Electrical activity in the axons. Realize that your neurons have a resting membrane potential of about a -70 millivolts, established by negative molecules inside the cell and very much from the use of the sodium-potassium pumps. And the membranes are permeable to the ions. The pumps maintain the potential. High concentration of potassium inside the cell, sodium outside of the cell. They can change their membrane potentials, and you call that excitability or irritability because of the change in membranes to certain ions. The ions will follow the electrical chemical gradient, moving from an area of high concentration and the attraction of opposite charge. The flow of ions is called the ionic current. At rest, a neuron is considered depolarized. So as the potential or the nerve impulse is moving along it, it's polarized when it's at rest. It's depolarized as the potential's moving along it, then it's repolarized, and for a little while it becomes hyperpolarized. This is a point at which it cannot fire again for a fraction of a second. Changes are recorded on an oscilloscope. Depolarization occurs when positive ions enter. Hyperpolarization when ions leave the cell. Usually it's potassium or chloride that enters the cell. Depolarization is excitatory. Hyperpolarization is inhibitory. Action potentials threshold is about a -55 millivolts. The voltage gated. Sodium channels open, sodium rushes in, the cell depolarizes. As it depolarizes, more sodium channels open, the cell becomes more and more permeable to sodium. What happens is it goes from a -70 to an almost a positive 30 millivolt, and that's your action potential. Your action potentials are really all or nothing. Once it starts, it goes. It's not... you don't get kind of an action potential or a half action potential. When an action potential starts, it goes. What happens is in increasing sensitivity, increasing pain, the frequency of the action potential goes up, and or more sensors are involved in sending that sensory input. At positive 30, the sodium channels close, potassium channels opens, and you start your repolarization. Resting potential is a hyperpolarization. It's an overshot, and it's a point where it can't depolarize again. Goes down to about a -85. Then it's established back to the resting potential, and then everything starts all over again. Let's see if this will play. Apparently not. Okay, so action potentials again are all or nothing. Stimulus intensity will make action potentials occur more frequently. So it's a frequency for your action potentials. This is called recruitment. Another time we'll use the word recruitment is when in muscle contraction, as we lift a heavier object, more muscle fibers will be recruited. That's another type of recruitment. Okay. Acetylcholine. Acetylcholine is a neurotransmitter, and it can be either excitatory or inhibitory. A neurotransmitter is what causes the ion channels to open. It binds to a receptor. It's also in the synapses between the neurons and carries the carries the depolarization from one neuron to another. It's excitatory in the CNS and some of the autonomic motor neurons. It's inhibitory in some autonomic neurons. You have two types of acetylcholine receptors, nicotinic, stimulated by nicotine, found in the motor end plates of muscle cells, autonomic ganglia, some parts of the CNS. You have muscarinic acetylcholine receptors, stimulated by muscarine from poisonous mushrooms. You know, as we talked in class, this is how, by adding exogenous nicotine or opiates or cannabinoids, this is why you're getting the high or the reaction you're getting, is because it's causing these nerves to fire. It's replacing this type of naturally occurring neurotransmitter with an exogenous one, or one from the outside. Agonist and antagonist. An agonist is a drug that can stimulate a receptor. Antagonists inhibit a receptor. Atropine would be an antagonist. Curare, as we talked about in class, is an antagonist. So it blocks the nerve transmission through the nicotinic receptors. We've talked about depolarization, potential hyperpolarization, the current, and another substance we look at is gamma-aminobutyric acid. It's the most common neurotransmitter in the brain. It's inhibitory, opening chloride channels, binds to its receptor. It's involved in motor control, degeneration of GABA-secreting neurons. In the cerebellum causes something like Huntington's disease, or called Huntington's chorea, or Huntington's dance. And this is an excited, when the gamma is not there, degenerated, you get these constant neurons firing because there's nothing to inhibit it. And these are these are diseases that you see with GABA. You'll see it in Huntington's. With other ones, you'll see it in Parkinson's. And if you see somebody with this, you see that they have some sort of neurological damage or neurological problem. This is just one on GABA, reflex arc. In skeletal muscles, when a muscle contracts, it always shortens. This places tension on a tendon or a bone to the bone, places tensions on the tendons connecting to the muscle to the bone. It moves the bone at a joint. The bone that moves is attached to the muscle insertion. The muscle attached to a bone does not move is the origin. So if it's your biceps, your insertion is at the lower end, your origin is at the upper end. Different type of muscle actions. You have an extensor that increases the angle of a joint. If you've flexed your arm, your triceps are the extensor. It increases the angle of a joint. A flexor decreases this. That would be your biceps. An abductor moves a limb away from the midline of the body as if you were lifting your arm. An adductor moves it back toward the midline. A levitator moves the insertion upwards. A depressor down. Rotator rotates the bone along its axis. A sphincter, such as the anal sphincter, constricts an opening. Single motor unit and the muscle fibers it innervates. A motor unit innervates muscle fibers. The contract strength comes from a motor unit. For finer muscle controls, you require more units controlling a small number of fibers. Such as in the eye, you may have less than 20 muscle fibers per motor unit. On the other hand, the large, strong muscles that don't need a lot of real fine control, you might have one motor unit firing 1,000 different muscle fibers. That's the difference between the control and strength trade-offs. Muscle fibers have densely packed subunits called myofibrils that run the length of the muscles, and they are thick and thin myofilaments. This is a video on sarcomere contraction, but I bet it's not going to play. Okay, you have cross bridges that form. Cross bridges, in order to form, need calcium. The regulation of the contraction, or the muscle contraction, the tropomyosin physically blocks the cross bridges. Troponin complex, troponin I, inhibits the binding of myosin. Troponin T binds with tropomyosin. Troponin C binds to calcium. And it regulates contraction. As you see, you have your troponin complex, your tropomyosin. Nothing's happening right here. When a muscle cells are stimulated, calcium is released into the muscle fiber. This attaches to the troponin C, causing a conformational change. This allows the myosin in to access to form cross bridges, and the cross bridges are what allow the muscles to contract. And again, this is probably not going to play on this. Okay, contraction of skeletal muscles. Studies done in vitro, one end of the muscle is fixed, the other is movable. Electrical stimulations have been applied. Muscle spindle apparatus. It contains thin muscle cells called intrafusal. You have nuclear bag fibers, nuclear chain fibers. You have two sensory cells wrap around the fiber, primary and secondary. Here's a picture of the muscle spindle apparatus. You have two types of lower neurons. You have the alpha, which innervate the extrafusal or the contracting muscle fibers. You have the gamma, innervate interfusal, the stretch muscle fibers. Contraction of these fibers does not shorten the muscle, but does increase the sensitivity to stretch. They are stimulated by upper motor neurons at the same time. It's called coactivation. Skeletal muscle reflexes. Skeletal muscles usually are referred to as voluntary and controlled by higher brain. They can contract unconsciously in result of certain stimuli, and that would be like a stretch reflex. A stretch reflex is what you would get if you tapped below the patellar or the patellar tendon. It only involves a sensory neuron synapsing on a motor neuron in the spinal cord, which means it does not go up into the brain, not up into the pyramidal tracts. It just goes to the spinal cord and back. Optimal resting length of a skeleton, it means the optimal resting length, stimulating by striking the patellar ligament, a knee-jerk reaction. Monosynaptic stretch reflex, stretches on a muscle, stretches spindle fibers, activates a sensory neuron. Sensory neuron activates an alpha motor neuron. Motor neuron stimulates extrafusal muscle fibers to contract. The stretch on the spindle is reduced, and you get your knee jerk. Here is a picture of it. Again, this is out of the book. Isotonic and isometric contractions. An isotonic contraction, the muscle fibers shorten when tension is produced, is just greater than the load. So if you're lifting a light object, you use fewer muscle fibers. If you're lifting a heavy object, you use more muscle fibers. Again, it's called recruitment. Now, isometric contraction, muscles can't shorten because the load is too great, and that can be voluntary. You have people that do isometric exercises. It helps strengthen the muscles. The muscles are not shortening. They're because the weight of the object, whether it's the chair you're sitting on, and obviously you can't lift yourself off the ground if you're lifting on the chair you're sitting on. That's the type of isometric exercise. Concentric and eccentric. Concentric reaction of muscle fiber shortens when the force is greater than the load. That's how you're allowed to lift. An eccentric contraction of muscle may actually lengthen despite contraction.